I'm Ben Temperton. I've been at University of Exeter now for about 8 years, I think, and my research is really looking at how bacteria and the predators of bacteria called bacteriophage kind of interact and evolve together. And one of the main angles we're trying to look at is how potentially these viruses of bacteria, and you can imagine viruses have got a really bad rap over the last couple of years, but actually the viruses that infect bacteria may actually become one of our strongest allies to treat another pandemic that's already with us, which is this pandemic of antimicrobial resistance and the fact that antibiotics are not working as well as they used to. So without further ado, I'll just go through the talk. Okay. So we're all here to talk about bacteria, essentially. Hopefully most of you are aware of what bacteria are. Typically when people listen about bacteria, they tend to think of them synonymously with the idea of germs. We're always taught about how, you know, Dettol kills 99.9% of bacteria, and that's a really great thing. Actually, the world is very much covered in bacteria, as I'll go on to in a minute, but they tend to be defined as these single-cell organisms, and they reproduce by dividing. They simply replicate their genome and then essentially bud off another bacteria. And some bacteria can do this very, very quickly indeed. So an average E. coli takes about 20 minutes maybe to make a new daughter cell that then immediately starts making its own daughter cells. There are others that can do it in 8 minutes. That's about as fast as it gets. So you can see how one bacteria can become many, many bacteria very, very quickly, which is why the argument for Dettol saying 99.9% of bacteria are killed is fine, except the 1% or the 0.1% you're leaving behind will pretty quickly take over that surface again. So it's a you know, it's great for that time where you've sprayed it, and actually the ones it kills sorry, the ones it doesn't kill are probably the ones you really wanted it to. So just be aware, if you're spraying everything with bleach, it will be clean for a relatively short amount of time after you've done that. Okay, so it's fair to say that bacteria aren't just the germs that we've kind of come to fear. We actually live in a microbial world. So pretty much every milliliter of seawater that you ever swam in has got about 1 million bacterial cells living in it. So, and these things are fundamental to what makes the Earth habitable, right? They are everyone hears this statistic where, you know, the rainforests are the lungs of the Earth, and they produce all the oxygen that we breathe. Actually, about half of the oxygen on Earth comes from marine bacteria that are also photosynthesizing. So huge amounts of bacteria live in the oceans. Soil has even more bacteria in it. You know, the density of bacteria in soils is even higher. They're hugely important in terms of nitrogen cycling and making it so that soil can actually, so the plants can get the nutrients out of the soil they need to grow. So if you get anything out of this, stop thinking of bacteria as bad. Bacteria are absolutely critical to making the world habitable. It's why they're going to Mars and looking for bacteria there, right? Because they think they'll probably be there as well as the kind of simple forms of life. But it's very clear that, you know, over 99% of species of bacteria are not harmful to humans. There are probably 10,000 to 100,000 species of bacteria out there. They are by far and away the most abundant kind of biodiverse format of life that we have on Earth. Within our body, we're covered in bacteria. Every part of our skin is covered in bacteria. Our guts are covered in bacteria. I don't think there's a single part of the human body that's sterile, really. If you remove the bacteria, you take out about 200 grams of your body weight would be just bacterial cells in there that are really, really, obviously you have to look at them under a microscope because they're so small, but it's, you know, a huge mass of single-celled organisms living in your body. These things are critical in your body in terms of making you know, vitamins available for you to uptake, and all these sorts of things is where bacteria come in. So almost all life on Earth has an associated microbiome with it that allows it to function successfully. So there's been a couple of papers where they've looked at this, where they've determined the number of bacterial cells to human cells in the body, and it's about a 1-to-1 of bacterial cells to human cells if you include white blood cells, and then if you exclude those, it's about 10 times as much bacteria as human cells. So it's, you know, if you think about the human genome, and people have sequenced that to look at it, well, actually, if you think about all the different bacterial types in your body, and they all have their own genomes as well, then actually the vast majority of the DNA in your body, in terms of the diversity of it, is bacterial. So, we've evolved with these things for several 100,000 years now, and we are, we're all one ecosystem, essentially. Okay, so it becomes a problem, though, when you have fast growing bacteria, or these typical things that can double in 20 minutes or so. That allows them to evolve very, very quickly. Now, by evolve, I mean they will pick up random mutations in their genome, and then selection will occur on those random mutations that will allow the ones with the right mutations to succeed. In the terms of antibiotics, what that means is that a particular bacterium will evolve a trait that allows it to survive the antibiotics being given to it to treat a patient. Now, there was an amazing video by a colleague of mine that he did several years ago that's available on YouTube, and I'll just play it for you now. It kind of shows how quickly this evolution can occur in a bacterial plate.
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, pour some thin agar so that bacteria can move around it. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
So it's worth a take home from that, remembering that it took 11 days for those cultures to go from being completely, susceptible to that antibiotic, they were killed by it, to becoming resistant to 1,000 times the original concentration of that antibiotic. Evolution in bacteria occurs really, really quickly. So where this becomes a problem is actually kind of how we've been using antibiotics for a long period of time. So typically you go home, you go to a doctor, you're given an antibiotic prescription, you take those antibiotics, and hopefully you're finishing the course of that treatment. But at the same time, some of that antibiotics will pass through your body and come out as waste. With animals, they found out long ago that actually you can make cows fatter quicker if you actually give them low doses of antibiotic, because it changes their microbiome and it allows them to take up nutrients quicker. So we've been dosing cattle for long periods of time with sublethal doses of antibiotic, which then passes through the cattle and goes into our water systems, and then basically collects there. That then meets kind of your bacteria that live all over the earth, and they start to have this selection occur on them to start finding ways to kind of get around that antibiotic. One of the clever tricks that bacteria can do is actually, if they develop these kind of mutations that allow it, they can then pass those on to other bacteria in their local community. So essentially they can develop a trait to become resistant to antibiotics and then package that trait up and then pass it on to not just their own kind of daughter cells, but also other bacteria in those regions. So antibiotic resistance can spread really, really quickly, and it has been doing, for many, many years. So we are now at the point where we have some bacteria that are now resistant to almost all of the antibiotics that we have developed since the 1940s when they were first discovered. Some of these names should hopefully well, they'll be in the news, not hopefully, but they will definitely be in the news. You might become more familiar with them as time goes on. So acinetobacter baumannii is the World Health Organization's kind of critical #1 threat. For this, there are strains of this that are resistant to every single antibiotic we have ever developed. And so if you get infected with this, there's sometimes been no treatment options available to you with the current antibiotics that we have. Pseudomonas aeruginosa is another one. We are now seeing strains that really are resistant to everything we have. MRSA is one that typically makes the news. That is a bacteria that's very resistant to antibiotics. And all of these three things typically tend to be a problem when you've been in hospital for a long period of time. Obviously, hospitals are very clean. There's lots of antibiotics being used, and so they are the perfect setting for bacteria resistant to them to kind of propagate, which is now one of the reasons hospitals really want you to go home as fast as you can, because it actually reduces the risk of picking up one of these infections. It's fair to say in the UK, infection rates of this are actually quite low at the moment, so don't be quite too alarmed. But it's definitely on the radar of kind of clinical stuff out there at the moment. So in other parts of the world, though, it's becoming a much bigger problem. So there's a paper out recently at the beginning of this year that actually showed that antimicrobial resistance is now the world's 3rd biggest killer after stroke and heart disease. So antimicrobial resistance is often talked about in kind of future terms of this. It's estimated that in 2050, for instance, there will be another 50 million deaths associated with antimicrobial resistance. But it's actually happening now in many parts of the world, particularly parts of the world that don't have well-developed kind of healthcare systems that can give you alternative treatments if you do become sick from these. So a lot of the countries in kind of lower middle income countries are really going to suffer the brunt of the antimicrobial resistance crisis as it starts to hit. So why are we in this position? Why don't we have new antibiotics coming that can treat this? Well, the reason is economics. It costs about $1 billion and takes about 9 years to get a new antibiotic to market. Typically, resistance to those new antibiotics emerges within 4 years of it hitting the market. So most pharmaceutical companies have now kind of abandoned doing any antimicrobial resistance work, simply the fact that it's very difficult for them to make a profit doing this for a long period of time. So they've been pulling out of it. So we basically need to start thinking of alternatives where we can either, #1, publicly finance research into novel antimicrobials and not leave it up to companies to do. But the other approach is actually to look at what did we do before we had antibiotics. So it's worth thinking that it was actually only really in the 1940s that antibiotics started to be produced on a mass scale and used. But long before that, disease has been around. So what did people do before antibiotics came to the fore? Well, one of the things they did is they actually used things called bacteriophage. So bacteriophage are, in this picture here, this kind of brown is the outside of a cell of a bacterial cell. And then these kind of cool spacelandy type things here are the bacteriophages that infect them. And I actually have a kind of model one here that you might be able to see. And essentially this binds onto the bacterial cell and then injects its DNA into the bacteria. And just like all viruses, just like COVID-19 does in the human body, it basically uses its own DNA to hijack the cell that it infects to make more copies of the virus. So Félix Darel was a French Canadian who discovered these things in 1917, and then they were independently discovered by Frederick Tort in 1915 in the UK. But Félix Darel kind of developed them into a medicine. So he figured out that if he had patients with dysentery coming into his clinic, and then he collected some of the samples that he would get from those patients, he could use those to treat other patients who also had dysentery, because the recovering patients would have lots and lots of these bacteriophages or phages, as we call them for short, in those samples. So he basically went to work on trying to figure out what the kind of active agent was in these treatments, and isolated these cool bacteriophages that essentially can cure these diseases. So this became known as phage therapy, as you can imagine. And so he set up this center in the Lavia Center in Georgia, in former Soviet Union, and they're basically still treating people with phage therapy to this day. So one of the things that I'm really excited about phages being a useful treatment of bacteria is essentially they have all the kind of perfect properties that you can imagine for any kind of medicine that you might want to develop. So the first one is that they are very specific, right? So phages will only kill the bacteria that they can inject into, and they'll leave all the other bacteria alone. So unlike antibiotics, which will cheerfully wipe out your microbiome in your gut and potentially can cause some side effects, you know, as your microbiome changes. This is often seen in people suffering from kind of long-term illness like C. difficile. The phages will actually only kill the pathogen that they're kind of, you know, evolved to kill. So they can be very, very kind of targeted medicines in that sense. The second one is what we call their autocatalytic. So it's every infection that a virus has on a bacterial cell will hijack that cell's metabolism and make more viruses. And typically for every bacterial cell that gets infected, it produces about another 50 to 200 new viruses or phages that then go out and kill the neighboring cell. So you can imagine you have to get relatively few phages to the site of an infection, where actually it'll start that cycle, and just like a kind of chain reaction, and a kind of, I guess, if you understand how fission works in nuclear reactors, it's exactly that. So you have one phage goes in, it produces multiple phage, they then go on to infect multiple cells, and it exponentially increases that way. So very, very few viruses that getting to the site of infection can actually pretty much clear an infection out. So they're very, very effective in that sense. The other nice thing about them being autocatalytic is actually once the infection is cleared, there are no new prey items essentially for them to hijack and make more viruses. So they stop replicating at that point, and then the human body just simply clears them away and clears them from the system. They are about as safe as any medicine as it's possible to have. So people have, many years ago, when you were allowed to do this without ethics, cheerfully drunk liters and liters of phage stocks without any known side effects to them. Phage therapy has been going on for about 100 years in like I say, in Georgia and other places. And there are very, very few cases where the human immune response really either doesn't recognize them at all and just simply ignores them, or it might mount a slight immunoresponse. So you might get some kind of, I guess, like a minor allergy reaction to it, at which point you can just simply reduce the amount of viruses you're giving to someone, and then that immune system stops panicking about it. So there, like I say, compared to some of the kind of last line antibiotics that we have that can potentially damage your liver and kidneys and these sorts of things, phages are about as safe as a medicine as it's possible to have. The other thing that's really super cool for it, especially if you think about potentially the impacts of antimicrobial resistance in low and middle income countries, is actually phages are phenomenally cheap in order to turn them into medicine. So when we isolate phages, typically going from the isolation all the way through to kind of genome sequencing and characterizing these things, costs between about 100 and 200 pounds to do that per phage, comparing it to $9 billion for an antibiotic. So you can scale these things up very cheaply. You can make it available to people with very, very few resources, and actually, provide this kind of global response to the antimicrobial resistance without it being controlled by very rich pharmaceutical companies. Okay, so where this came into play is essentially when Felix Doral started this. They really started developing these things as medicines. I just want to reiterate that this has been being used for a very, very long period of time. So when he set up the Alavi Institute, they used to find troops in the Second World War with these little capsules on the right hand side that are actually full of phages that have been isolated from bacteria. And what the troops used to use it is if they were hit by by a bomb or something, or shrapnel, and had a wound, they would pour these phages into the wound in order to stop bacterial infections from taking place. And these are all developed in the Alavi Institute, which is the building at the bottom that is, again, still practicing to this day. Probably one of the world's greatest centers of phage therapy. But then, so why did we stop using them? Well, Alexander Fleming came along and he invented penicillin, or discovered penicillin, in, I think, 1923-ish. And so then after it was made into a drug, they suddenly had a drug that wasn't specific. You could actually, a patient would come to you and you could just give them antibiotics regardless of either whether you knew that they had a bacterial infection or not. And if you certainly didn't know what the type of bacteria was that was infecting them, the antibiotics they would give you would treat that infection. So it's kind of seen as a silver bullet where you could rapidly produce a drug that could treat all these different types of bacterial infections without necessarily having to worry about identifying the type of bacteria that was affecting it. So they were definitely kind of seen as kind of the silver bullet approach to this. So at least in the West, where they had access to antibiotics, phage therapy kind of really died out. They really stopped using it. Phages still were useful biologically for understanding kind of fundamental biology. So the fact that we know that DNA is the genetic material comes from work done on phages. But it was only really in Eastern Bloc countries where they didn't have access to these antibiotics that they continued to develop phage therapy and progressed it in that way. All this kind of came back to haunt us really when the antimicrobial resistance crisis started to be made, that it was clear that it was here and causing a problem. And in the West, really the catalytic story for this was this amazing story by Tom Patterson here and his wife Stephanie Strathy. Now, Tom Patterson was Stephanie Strathy's husband. They were on a retreat to Egypt to celebrate their wedding anniversary, I think, and he became sick. He had a gallbladder infection, and he was infected by this strain in the right-hand corner called Acinetobacter baumannii. Now, as I talked about before, Acinetobacter baumannii is extremely resistant to antibiotics, and so they couldn't find an antibiotic that would clear this particular infection. So he was rushed back from Egypt first to Germany, and then he went to the United States. And again, they couldn't treat him there, even though using the last line of defense medicines. So his wife, who was an HIV researcher, actually hit the literature and started figuring out what people did before antibiotics, got in contact with all of these kind of phage biologists that happened to have these collections of phages, and asked them if they had any phages that would kill Acinetobacter baumannii. And sure enough, they did. And so they sent them collections of them, and they tried putting this in Tom Patterson, and essentially they cured him. They cleared the infection up. And I think after 9 months of intensive care, he actually came out and he was fine. The infection was cleared. And if you like reading cool science literature that have kind of true stories, there's an amazing book called The Perfect Predator that Stephanie Strathy wrote that kind of describes this. I spoiled the ending for you. He lives, but it's a really cool story about kind of how kind of her diligence and how they continue to kind of push against kind of, find new options when he'd run out of other options. They also set up a new phage therapy center in San Diego in the US following the success of this. So that's called IPATH, and it's still working at the moment. Okay, closer to home in the UK, there has been one recorded case in hospital where phages have been used to treat a patient that was untreatable. And this was Isabelle Carnell-Holdaway. She was a 15-year-old girl. She was at Great Ormond Street Hospital, and she had cystic fibrosis. And they basically needed to give her a double lung transplant so that she would survive. So they did that. And then during that, she got infected. She got infected by something called mycobacterium abscessus, which was pretty much resistant to everything. And that infection spread through her body. And she was basically sent home for palliative care age 15 because they couldn't treat her. So the clinician wrote to a guy in the United States who had a collection of 18,000 phages that infected a similar strain of bacterium, and 3 of those were found to actually infect the strain that was infecting this particular girls. So they brought those over from the States. They had to do some genetic engineering on them to make them safe to use in people. But after that, they treated her, and sure enough, she got better. And I think a few years ago now, she sat her A levels and is doing great. So it's, a true success story in the UK of where phage therapy has been used to save somebody's life. So where do these phages come from, right? Where do we find them? Well, the truth is phages are absolutely everywhere. Wherever you find bacteria, you will find phages. And it's estimated there are around 10 to the 31 viral particles on Earth, right? By far and away the most abundant ones. So that's basically a 10 with 31 zeros after it. So a huge, huge number, which is still mind-blowing to people. We just can't understand numbers that big. So let's put it this way. If you think about a virus being on average 200 nanometers in length. Now, a nanometer is basically the speed, it's the amount your nail will grow in 1 second on your hand, right? So it's a tiny, tiny amount. So phages are basically, if you thought about 200 seconds passing by, that's the length of your nail that it would be after 200 seconds. That's a single viral particle. Well, if you stack them end to end, then actually the whole thing would span about 200 million light years worth distance. That's what 10 to the 31 means in terms of numbers. So we are in this kind of local galactic group here. This is known as the Virgo supercluster. Down here in the Virgo cluster is where they took that picture of that black hole several years ago. And so you would go across this whole distance and back again if you stacked all the viruses. So phenomenal amounts of viruses out there just waiting to be discovered. And what we rely on in the Citizen Phage Library, and it seems to be the case, is that pretty much every drug-resistant bacteria out there somewhere has a phage that will kill it. And all we need is to find that phage and then turn it into medicine so we can treat these different types of disease. So how do you do that quickly, and how do you scale it up and make sure that it's all ready to do when the antimicrobial resistance comes knocking? So the best way of doing that really is through citizen science. So that's why we set up the Citizen Phage Library. And the idea was essentially that citizens like yourselves would collect samples. They would go out and collect freshwater samples. They would then identify on where they got those from using the What 3 Words app, which essentially just puts your location where you are on Earth. And then we send those back to the lab for processing. Now, because viruses are everywhere in the environment, you can essentially find viruses in fresh water that will affect and kill these drug-resistant bacteria that are out there. So we get them in, we isolate them. We basically, once we have the viruses, we screen them to make sure that they don't have any genes that we really wouldn't want to be putting in a person. So we make sure they don't encode new antibiotic-resistant genes. For instance, that would make everything worse. So we avoid those. Almost all of them don't. And then once we have that information, we sequence it. With that way, we know we have a novel virus, and most of the viruses we find are new. I'll talk about that briefly in a minute. We collect all that data into a database. And at that point, clinicians around the world can then say, we have a patient in our hospital that is infected by a Pseudomonas aeruginosa. Do you have any viruses in your collection that might be useful to treat this particular patient? And they can look in our database and identify the ones that might be useful and get in contact with us, and then we can test them on their strain. The nice thing for the Citizen Phage scientists is that actually once the phage is found in those samples, then you get to name it, right? So you will become the discoverer of that phage in all the scientific literature. So you'll become a real kind of long-term kind of record of your efforts to make this happen. We've had some very cool names. Kids come up with the best names for naming these phages. So I'll talk about some of those shortly. So when we get them back to the lab, the first thing we essentially do is we filter out any bacteria that are in those samples, and then we add to them any particular bacteria that we're looking for phages for, right? So this might be a clinical strain of a bacterial pathogen that's infecting a patient. If there are any viruses in that water sample that infect that particular bacterium, they will attach to it, they will inject their DNA, make more viruses, and then amplify that system overnight. Once we have that, we basically then plate that out onto a lawn of bacteria, and that's what you can see on the right here. So on this lawn, the kind of cloudy yellow stuff here, the kind of beige, is the bacteria that are growing on the lawn. And hopefully you can see that in some places there are these holes where we've put the samples that we've isolated from there. Now, what those holes are caused by is a virus has infected a cell at that particular point, made more viruses, and then spread outwards as it killed that bacterial lawn. So you get these zones of clearing that are called plaques. And what we're looking for is these plaques. And you can see how some of these plaques have different features on them, right? So this one is really, really clear, and it has this kind of halo effect going on outwards. Now, these are what we really, really want, because it shows that they're killing them very, very well. That's this clearing in the middle, and they're also producing compounds that are, even when the phage has stopped growing on this lawn, it's still, those compounds are still weakening the bacteria beyond that. So these are really, really good ones. Sometimes you can see we get these cloudy points here, like sample 15 we put on. Basically what that means is initially the virus killed that particular bacterial cell at the line, and then the bacteria grew over the top of it. So the bacteria became resistant to those viruses, just like they become resistant to antibiotics, and then it managed to grow back over the top of them. And then some of them don't yield any phages at all. There just weren't any phages in that particular sample that affect this particular strain of bacteria. So we've done this now for about 300 samples from across the Southwest. So you can see where we had these, the green pins and red pins are where we've taken samples in our current system. And from that, we've actually pulled out around 200 phages that we've managed to isolate from this. So it's actually a pretty good return rate from citizen science libraries. Right? The likelihood of finding a phage in your sample is pretty high. We've got some great phages from there that have actually been gone on to be used in, that work against real pathogen threats that are currently out there. So this phage here, acinetobacter phage lemonade, was discovered in the River Lemon. It was actually the first one that we found in the Citizen Phage Library. So this affects Acinetobacter baumannii, that same strain, the same bacteria that was infecting Tom Patterson in the perfect predator case. This one kills it absolutely stone dead. So it's a really good virus for killing those. We have a phage called Pseudomonas phage sedgwick that we've actually recently sent off to a clinic in Switzerland to be used for a patient out there. And again, this is a really, really strong killer. We had 2 phages that infect bacteria that were isolated from a citizen scientist's chicken coop, where we basically took a sample from there. And the initial one was found. We asked them for a name, and they called it Kylie Mineg because it came from a chicken coop. And then we found a second phage in there that became Danny Mineg. And they're actually different viruses from the same sample. And again, these are really strong, potent killers of Pseudomonas aeruginosa, which is another one of the kind of the threats. So where this really came to the fore is we had a request come in from a clinic in Lausanne University in Switzerland. There's a company set up called Phage Directory who actually kind of directs these requests for patients who need viruses that will treat them because they're antibiotic resistant. So again, this was a child with cystic fibrosis. She had a PAM-resistant strain of Pseudomonas aeruginosa, which was this horrible black-growing bacteria on the left-hand side. They screened 140 of their phages that they had in their own collection, and none of those were successful at killing this particular strain. So 8 labs globally answered the call and basically said, right, we'll start screening our phages for this particular strain of this bacterium. We received the strain on the 4th of November and then started the screening on the 5th of November. And within 3 days, we'd found phages that killed this clinical pathogen, suitable for treatment. We then sequenced it 6 days later because we bring all the sequencing in-house so that we can do this. And then we basically, we've recently sent that off to them so they can start using it to try treating that patient to see if it works well. So it showed that actually having these phages on tap from citizen samples really, really does yield very positive results for bringing these things forward. So hopefully I've convinced you that this is a good project to be in, and that your phages that you find in your samples may actually be useful. So we have currently, what we're going to be doing with your samples is we have a patient actually at the Royal Devon and Exeter Hospital who we are trying to get compassionate therapy for, because essentially the antibiotic regimen she's currently on isn't really working very well, and there aren't other options for her. So she really fits the bill for compassionate phage therapy. So we have found some phages against this particular strain in the past, and we could really do with more. So we're going to be using those to isolate phages and then navigate the ethics that is using phage therapy in the UK, so we can actually treat this patient with them. Once we get your samples, you'll be notified that we've got positive hits in there, and then you get asked to submit a name. So this is when you get the email saying, congratulations, a phage was found in your sample. Please start coming up with sensible names for them. They can be very creative. They typically have to avoid anything copyrighted. So it can't be the name of a film or something. Well, assuming it can be the name of a character in a film as long as the film wasn't named after that character. So you couldn't have Iron Man as your phage, for instance, because there is an Iron Man film. But you could have Hermione Granger phage because there isn't a film named after Hermione Granger. So be as creative as you like. Try and keep them relatively punchy. I had a PhD student once who was obsessed with Norse mythology, and now we have a load of phages that I can't pronounce, and I have to go and talk about them at conferences, and it becomes rather challenging to pronounce some of these ancient Nordic names. So it's just to bear that in mind when coming up with these names. And then now we have a potential name for your phage, and then what we do is we send it off for sequencing, and we compare the genome of the phage that you've discovered against all other phages that have been discovered previously. And assuming it isn't the same as something else, then actually the name that you chose will be assigned to it. Now, so far, I think we've found out of those 200, maybe one phage that was previously identified somewhere else in the world. So there's so many viruses out there that the likelihood of finding the same one twice is incredibly low. So you have a very good chance that your name will stick and be used in scientific literature if and when that phage gets used for therapy. Once it's in our database and it's classified and we've characterized it fully, then that will then be made available to clinicians around the world to treat others. So we are currently, I think we have 4 ongoing requests currently for various different bacterial diseases that we are now helping to kind of supply them with phages in order that their patients can be treated. And almost all of the phages we're sending have come from citizen science samples. So it really is a good way of getting help to people, not just in the UK, but also in other countries. Okay. Finally, just if you want more information about Citizen Phage Library, please sign up on the website, and then we'll basically notify people through that as kind of the stories progress and we do kind of pursue the project further. We are probably going to be starting a crowdfunding campaign at some point this year in order to just try and keep people employed and keep the project rolling as we navigate UK research bodies for funding that will fund this project going forward. So that's the website for the Citizen Phage Library. So please sign up if you want more information. And that's me. Are there any questions?
That was absolutely brilliant. Thank you so much, Ben. It's such an exciting project. So amazing that people can really make an impact through it, and also just exactly how science should be done. It's so open source and wonderful, and that's one of the reasons I've just found this incredibly exciting, because so often science is it's not done this way. It's done about profits, and this, as you've shown, is it's cheap and it's great and it's accessible to everyone, and it's so exciting. So thank you very, very much.